My name is Dr. Mark Pilgrim, and I've created this photo story to introduce you to selective and differential media and the interpretation of that media. Selective and differential media can be used to both isolate and differentiate groups of bacteria. In the microbiology lab, we use a variety of these media to study microbes. Selective media will contain chemical agents that allow some microbes to grow while inhibiting the growth of others. For the microbes that grow, we say the microbe tolerates the selective agent, and the media selects for that microbe. Differential media will contain chemical agents that create a visible difference in some microbes, allowing them to be distinguished from others. Differential agents do not normally affect the growth of microbes. Many media are both selective and differential. Not only do they allow certain microbes to grow, but there is also a visible difference among those that grow that indicates a specific sugar is fermented, or an ingredient in the media is used, or if a particular enzyme is present. This really depends on the medium. After media has been inoculated and incubated, you will record and interpret your results. It is not simply enough to describe what you see, you must tell others what it means. This is called interpretation. This is a general scheme for interpreting selective and differential media. There are three basic steps. Number 1, observation of growth. Number 2, observation of a change to the media. And number 3, identification of most likely microbes or microbe. Not all steps are used for all media. When the media is selective, you will use steps 1 and 3 only. If the media is differential, you will use steps 2 and 3 only. If the media is both selective and differential, you will use all 3 steps. When observing growth, you will determine if there is good growth or if the growth is inhibited. You will determine what is good growth by comparing your unknown result with a positive control, an organism known to grow well on that media. It is also helpful to include an organism on the plate that is known to grow poorly or not at all on that media as a negative control. If there is good growth and the media is only selective, you can proceed to step 3. If there is good growth and the media is also differential, you will proceed to step 2 to interpret the differential part. If there is poor growth or no growth and the media is also differential, you cannot interpret the differential part. Good growth is required to interpret the differential part. Typically, the differential part of media will exhibit a color change of some type to distinguish between various groups of bacteria. This will vary according to the medium. If there is a change in the medium, usually in color, the organism is considered positive for whatever activity the medium measures. This activity will vary according to the medium, and so will your interpretation. You would then proceed to step 3. If there is no change in the medium, the organism is considered negative for whatever activity the medium measures. You would then proceed to step 3. On step 3, you may be able to name the genus and species of the microbe, but most of the time, one medium is not sufficient to do this, and your interpretation will be more general in nature. For example, the microbe is most likely gram-positive, or the microbe is most likely a coliform. Your statement for step 3 will vary according to what medium is designed to measure. Let's look at a few commonly used selective and differential media and practice interpreting some examples of results. The first selective and differential medium is MacConkey agar. You should note that the selective and differential agents are listed on this slide. Its purpose is to isolate gram-negative enterics and to differentiate them based on their ability to ferment the sugar lactose. On this MacConkey agar plate, you can observe there is no growth. Therefore, the interpretation would be, microbe did not tolerate bile salts and crystal violet, and is most likely gram-positive. You cannot interpret the differential part because there was not good growth. Here are 2 examples of good growth of non-fermenters. Your interpretation would be, microbe tolerates crystal violet and bile salts, does not ferment lactose, and is most likely a non-coliform. In this example, you should observe bright pink or even red colonies. Note the bile salt precipitates in the media surrounding the colonies as well. Your interpretation should be, the microbe tolerates the crystal violet and bile salts, it ferments lactose, and is most likely a coliform. The next selective and differential medium is eosin methylene blue agar. you should note the selective and differential agents listed here. Its purpose, just like MacConkey, is to isolate gram-negative enterics and to differentiate them based on their ability to ferment the sugar lactose. You'll notice in this picture of EMB that the medium is one consistent color. There is good growth, but no color change. Your interpretation would be, the microbe tolerates the dyes present, but does not ferment lactose. The microbe is most likely a non-coliform. Notice the darkening of the growth on this EMB plate. The interpretation would be, microbe tolerates the dyes present, ferments lactose, and is most likely a coliform. In this example of EMB, you see a striking green metallic sheen on the medium. This unique coloring comes from microbes that ferment lactose vigorously, such as E. coli. Your interpretation should be, the microbe tolerates the dyes present, ferments lactose, and is most likely a coliform. Mannitol salt agar is both selective and differential. Again, note the selective and differential agents listed here. The primary purpose for this medium is to isolate gram-positive microbes, especially Staphylococci, although a few other gram-positive microbes will grow on this medium. Color change in the medium to yellow will indicate a mannitol fermenter, whereas no color change will indicate a non-fermenter of mannitol. In this example of mannitol salt agar, no growth is seen. If you look carefully, you will see the streaks left by the loop, but there is no growth. Interpretation would be, the microbe cannot tolerate salt and is most likely gram-negative. In this striking example of mannitol salt agar growth, you can see a dramatic change in the color of the media. Your interpretation would be, the microbe tolerates the salt, it ferments mannitol, and is most likely gram-positive. The most likely identification in this case might be Staphylococcus aureus. Interpretation of these results on mannitol salt agar would be that the microbe tolerates the salt, but does not ferment mannitol and is most likely gram-positive. You could go further and say that it is most likely a non-aureus Staphylococcus species. Another medium typically used is phenylethyl alcohol agar. It is selective only. It contains a selective agent, but no differential agents. It allows certain gram-positives to grow, but inhibits most gram-negatives. Since it is not differential, there is no color change associated with certain groups of microbes. You can see that several microbes have been used in inoculating this medium. Good growth can be seen in the lower left and upper right-hand quadrants. Poor growth, that is mostly inhibited, can be observed in the upper left and lower right quadrants. For good growth, interpretation would be, the microbe tolerates the phenylethyl alcohol and is most likely gram-positive. For the poor growth, interpretation would be, the microbe cannot tolerate the phenylethyl alcohol and is most likely gram-negative. I hope that this photo story has been helpful. If you have any further questions, you may contact me by the email listed on this slide.